Henry Rutgers.
Recording in progress.
Henry Rutgers Chair of the Human Microbiome. He also is a professor of medicine and pathology and lab medicine, and also, obviously, one of his main roles is as director of the Center for Advanced Biotechnology and Medicine. Marty, in my opinion, was a big reason why I even considered looking, and has become a mentor to me, which I appreciate a great deal. So without further ado, Marty, take it away.
Right. Yeah, I hear an echo.
I hear an echo.
Do we know how to, firstly, can you see my screen?
Yes. Yes.
Okay.
Yeah, if everyone can mute, maybe the echo will go away. So, so Keith, thank you very much for your kind introduction. Colleagues, I'm a professor at Cabham across the river, but I'm also a professor of medicine in the Division of Infectious Diseases. So I'm your colleague, and my whole career has been in infectious diseases since I started my fellowship in 1977. So that's 45 years. And I love the specialty of infectious diseases. So today I'm going to talk about something that has a kind of a change in my way of thinking that has developed gradually over the last 20 years. And I call it the double-edged sword of antibiotics, climate change in miniature. And I think it is likely that I'm going to show you some slides and some ideas that you've never seen before, which is great. And also, I'm very happy to be interrupted at any point in this talk. So let me begin. So here, this slide shows 4 different things. A volcano erupting. And can you see my mouse?
Yep.
Oh, thank you. A volcano erupting, an earthquake, the formation of a mountain range, and an island archipelago. Now, you might think that these are all unrelated events. And the question is, what caused all these things to happen? And we now know that they are all related. They all have a single cause, and that is best summarized by the concept of plate tectonics, continents in motion. And it is the collision of the plates that results in all of these phenomena. So this is a theme. Unrelated things may have a single underlying cause.
How to reduce this volume? God Lord.
Am I talking too loud?
Nope, if everyone just needs to mute their lines.
Yeah, there are people who have not muted their lines. Yes,
Yeah.
please, everyone, mute your lines.
Okay, so one of the themes that I've been interested in is a question that we all know. Why have many diseases increased in recent decades? Shown here are 3 diseases: reflux esophagitis, juvenile diabetes, and asthma. All have increased dramatically over the last 50 or 70 years. And to that, we might add obesity, IBD, celiac disease, food allergies, atopy, ADHD, autism. So if 10 diseases are going up more or less simultaneously, do they have 10 different causes? Or perhaps there is a single cause underlying them all. And that's what I'm going to discuss today. So let me just say a couple words about obesity. Here we're looking at time trends of overweight and obese children all over the world. And we see that in developed countries like the United States, the curve is going up. We see in developing countries that the rates are lower in each year, but gradually they're going up too. And in fact, if you look at where the developing countries are now and where we were then, it's about the same. It suggests that the same thing is going on there as here, but it's lagging about 30 years. Those are the rates, but here are the number of overweight and obese children in the world. And this shows a somewhat different story because most of the overweight kids in the world under 5 are in developing countries because that's where most of the children live. So in fact, we're not just talking about an epidemic in countries like the United States, we're actually talking about a pandemic. It's slow and it's gradual, but its scale, as of 2020, there were about 50 million overweight and obese children under the age of 5. So this is gradually overweight and obesity is becoming more of a problem in developing countries than malnutrition. So how can we explain this? I want to start by talking about the microbiome, which is, I think you'll find not surprising coming from me. This is a study that was led by Nick Boklich when he was a postdoctoral fellow in my lab. It's a study of 42 children born in New York. And here we're looking at the microbiome in these kids by month of life. And on the y-axis is relative abundance. What's important is not necessarily the names, but really the colors. So children begin life dominated by proteobacteria like E. coli, and then gradually these wane to become a small proportion of their microbiome. And then we have the development of bifidobacteria and Bacteroides and Faecalibacterium and Clostridium. This is, in fact, a very orderly succession. And if you look at children anywhere in the world, you will see a very similar pattern. So this is not accidental at all. This is actually very conserved in human evolution. And in fact, we see the same thing in non-human primates as well. So where do we get these organisms? Well, it turns out that mom is very important. We humans are mammals. We're born in a womb that is essentially sterile. Our first exposure to the world of microbes happens when the water breaks. The baby descends through the birth canal and is covered by the mom's microbes. The baby swallows the microbes. Now there's skin-to-skin contact, mom and baby. The baby's mouth full of microbes inoculates the breast. And now microbes and substrate go in to form the foundation of the microbiota of the GI tract. Moms are kissing babies, licking babies, pre-masticating foods, lots of redundant pathways to transfer the microbes from one generation to the other. This is what happens in all mammals. And this is what's happened in all humans until recently. But now moms are different. They live in a world of antisepsis, antibiotics. They receive antibacterials in their diet. Babies are different too. They may be born by C-section. They miss the trip through the birth canal. In the US, 1 baby out of 3 is born by C-section. Babies are bathed extensively. They're bottle fed. They're given formula that only superficially resembles human breast milk. And they're getting lots of medication, especially antibiotics, which I'm going to focus on. So on the basis of these ideas, over the last 20 years, I've formulated an idea called the theory of the disappearing microbiota. And that theory has 2 major tenets. The first is that changed human ecology has altered transmission and maintenance of ancestral microbes, which affects the composition of the microbiota. And the second point is that microbes, both good and bad, usually acquired early in life, are especially important since they affect a developmentally critical stage. So over the rest of this talk, I'm going to show you some evidence that supports this theory. Now, about 10 years ago, Stan Falco and I expanded this hypothesis, which is shown here, the effect of maternal status on the resident microbiota of the next generation. Our idea was that ancient moms would transmit an ancient microbiota to their babies. But if they lost microbes and couldn't get them back horizontally, then the next generation would be born at a deficit, and so on and so forth. And in this model, the changes in the microbiome are cumulative across generation. Now, unfortunately, there's more and more evidence that this model is correct. I'll show you 2 examples. Here, we're looking at the prevalence of Helicobacter pylori in 3 generations of Japanese families. Until recently, Helicobacter pylori was the dominant organism in the human gastric microbiota, and it's stepping down, just as we anticipated. Now I'll show you data from looking at the overall diversity of the microbiota, the alpha diversity of the intestinal microbiota in 4 populations. This was a project led by Maria Gloria Dominguez. I participated as well. This was a study of healthy children in the US, in villages in Africa and in Latin America, and in an uncontacted group of Amerindians in South America. And the y-axis is the amount of diversity. What we see is this group of children has the highest diversity. This group has the lowest diversity. And we can almost imagine the step down with acculturation. There have been several other studies of different societies that show this exact same model as well. So we have evidence that we are losing microbial diversity. So where is it coming from? Well, it's coming from many places, but I'm only going to focus on one today, which is antibiotics. This is what we do for a living. Antibiotics, everyone recognizes, both professionals and lay people, as one of the greatest discoveries of the 20th century. Introduced in the late 1940s, saved innumerable lives, and have revolutionized almost every aspect of medicine. But as a result, health practitioners are using antibiotics more and more and more. How much more? Recent survey indicated greater than 73 billion antibiotic doses used worldwide every year. That's more than 10 pills for every man, woman, and child on Earth, and the numbers are going up. In 2018, the CDC calculated about 250 million courses of antibiotics were prescribed. That's 763 courses per 1,000 population. That's 3 courses for every 4 people. And this has been going on year after year. Based on the CDC data, in children, by the time they're 2, they've had nearly 3 courses of antibiotics. By the time they're 10, 10 courses of antibiotics. And pregnant women, more than 50% are treated with antibiotics or given antibiotics for prophylaxis. This is in the US. And there are exposures from antibiotic use on the farm. We don't even know the magnitude of this. Now, recently, I've teamed up with 3 anthropologists to survey to try to understand why there is variation in antibiotic use and why there's overuse of antibiotics. And this was published about a year ago in Bioessays. And some of the next few slides I'm taking from this paper. But for people who want to have an in-depth look at this, I recommend this paper. And this is, I'm going to focus on 3 recent papers that we've done. This is one of them. So first, let's go back to some older data. This is rates of antibiotic prescribing for sick children in 25 pediatric practices. This is a study led by Jeffrey Gerber at the University of Pennsylvania. It looked at 25 pediatric practices, all affiliated with the Children's Hospital of Pennsylvania, one of the best children's hospitals in the world. And here on the y-axis is the rate of antibiotic prescribing per capita, comparing these 25 practices. And between the practice that prescribes the least and the practice that prescribes the most, the amount of variation is 100%. And for broad spectrum antibiotics, the amount of variation is 400%. And these are academic practices, and the differences cannot be explained by the patient mix. This reflects differences in culture. Now, here are data from the CDC looking at antibiotic prescribing, outpatient antibiotic prescribing per 1,000 population per state. In 2018, I told you that the national rate was 763. In New Jersey, we're 844, we're higher than the national rate. And you can see that the colors indicate differences in the rates of antibiotic prescribing. And there are very strong regional patterns. So these states here with plus signs, all of them have greater than 1,000 antibiotic prescriptions per 1,000 populations per year. That's more than 1 prescription per capita. And in contrast, if you look at these states in the West, it is about less than 500 prescriptions a year, half the rate. Now, this is across tens of millions of people, and these rates have been stable for many years. Again, the extremes are West Virginia, 1185, and Alaska, 451. These cannot be explained by differences in the rates of serious bacterial infections. These reflect differences in practice and culture. Another interesting phenomenon is that antibiotic prescribing is much greater in females than males, about 50% higher in females, and we can discuss the reasons for this as well. Now, interestingly, it's interesting to compare this map of antibiotic use and this map, which is COVID-19 vaccination rates. It's almost the opposite. And that provides a lot of food for thought as well. Now, here we're going to look at 3 maps, and this was in our paper. This middle map was in 2010, antibiotic prescribing rate per 1,000 population. You see the same kind of distribution that you saw in 2010. And I'm comparing it with 2 other maps that show similar phenomena. Anybody have any idea what these two maps are? Well, this map is opiate prescribing per 1,000 population. A lot of similarities. And we might ask, why is antibiotic prescribing similar to opiate prescribing? And this map is not about prescribing, it's about church attendance in the United States. The states with higher antibiotic prescribing also have high rates of church attendance. Now, this may be completely irrelevant. There may be no relationship at all, but I'll show you some ideas that suggest that there is a relationship. And I'll just tell you now, people who go to church believe that there is a higher authority in life. That's why they have religious practices. And in a way, doctors are a kind of higher authority too. And antibiotics are a higher authority. And so it's not shocking that there would be a correlation. Now, let me quickly turn to what are the antibiotics that are prescribed in the United States so broadly. And the largest antibiotics in outpatients are divided into 5 classes: penicillins, macrolides, cephalosporins, increased spectrum beta-lactams, and tetracyclines. You see they're all in the tens of millions of doses. Amoxicillin is the leading agent, 164 prescriptions per 1,000 populations. Azithromycin, the Z-Pak, is given in essence to 1 in 8 people every year. Cephalosporins, including cephalexin, augmentin accounts for all the increased beta-lactams, and doxycyclines. These are the widely prescribed agents. Now, antibiotic prescribing, as I showed you in the pediatric practices, is not uniform. Here are data looking at cumulative percentages of antibiotics prescribed by Medicare Part D prescribers. These are in older people, according to prescribing volume and rates among high and low prescribers. The x-axis goes from 0 to 100. These are all the prescribers in Medicaid Part D. And the y-axis is the cumulative percentage of antibiotic volume prescribed. The top 10% of Medicare prescribers prescribe more than 40% of all the antibiotics. And the bottom 40% prescribe somewhere about 15% of all the antibiotics. So you can see that the prescribers make a big difference. There have also been studies of prescriptions in VA populations, and this does not reflect differences in patient population. It reflects differences amongst the prescribers. Now, here's another way of looking at variation. Here's variation per capita antibiotic prescribing in 31 European countries, ranging from the Netherlands at the low to Greece at the high. In general, there's a north-south relationship, that the northern countries in Europe are prescribing less than the southern countries. There is not a threefold difference in the rate of serious bacterial infections. This reflects differences in practice and culture. Now, a very interesting study on the sociology, the dimensions of culture in medical practices was published about 10 years ago by a European social scientist named Hofstede. And they talked about 6 different concepts about prescribing: the power distance, about the inequality between prescribers and their patients; uncertainty avoidance, the extent to which a society tolerates uncertainty and ambiguity; individualism versus collectivism; masculinity, masculine values, macho-ness; long-term versus short-term orientation, indulgence. And what they found in this 2009 Eurobarometer study, the strongest association with antibiotic use had to do with uncertainty avoidance. Cultures that had that liked certainty liked to prescribe antibiotics. Cultures that had high machismo, the going to work and going back to function, using antibiotics with work, these were the 2 factors that were most associated with high antibiotic prescribing. Now let's turn a little also to developing countries. This study, this slide is from a study of 8 urban populations studied by the Bill and Melinda Gates Foundation. Here we're looking at age in the first 2 years of life. These are kids, antibiotic courses per person year. This line in blue is a line that I put in. This is the rate of antibiotic prescribing in the United States. And what we see is that in these developing countries, in 6 out of the 8 countries, the rates of prescribing are higher than in the United States, including in Bangladesh and Pakistan, where the average child in the first year of life is getting more than 10 courses of antibiotics. Now, you might say, how is this possible? But these children have parents who love them. The child has a cough or a fever, and they go to a local pharmacist who, without a prescription, is happy to sell the parents an antibiotic. So use in developing countries, in many cases where the formularies are unrestricted, is greater than in developed countries. Not surprisingly, antibiotic resistance is also getting to be a big problem. And so here's studies from WHO, proportion of patients with bloodstream infections caused by E. coli resistant to 3rd generation cephalosporins. The rates are higher in low and medium income countries than in high income countries. We can look at bloodstream infections by MRSA. We see the same phenomenon as well. Very interesting work has come from China. Here's a paper looking at antibiotic levels in drinking water in Chinese children in Shanghai and their contribution to antibiotic exposure. The investigators looked at antibiotics in drinking water where they found it, and antibiotics in the urine from children where they also found it. And what was interesting is that in China, there are antibiotics that are approved for human use, they're approved for animal use, and there are antibiotics that are approved for both. And in the drinking water and in the urine from children, they found antibiotic levels that come from antibiotics only approved on the farm, indicating that there is some spillover of those antibiotics into the human population, either from drinking water or from foods of animal origin. Here's just one graph from that paper, frequency distribution of the daily dose estimates for all antibiotics in 518 school children. Basically, they couldn't detect antibiotics in 21% of the children, but they did detect antibiotics in 78% of the children, usually at low level, but in some cases at much higher levels. So at least in China, there's evidence that antibiotics, not only those given by prescription, but those from the environment, especially from the farm environment, are ending up in people. And this is especially important in children. This is just a summary. More than half the children were exposed to antibiotics around greater than 1 microgram per kilogram per day level. These are low levels, but in fact, we don't know what's the significance of being exposed to antibiotics day after day with these low levels. So many people are interested in the ecological effects of antibiotic exposure. And I draw this like the proverbial iceberg. The tip of the iceberg is antibiotic resistance. We've known about this at least since 1945. There's a lot of interest in this, but I believe that the body of the iceberg is what antibiotics are doing to the microbiome. They're disrupting it, and that has potential clinical consequences. The consequences, the disruption could be transient or long-term. The consequences could be of developmental, situational, senescent, or generational type. And they can affect immunological, metabolic, neoplastic, or maternal phenomena. I'll show you some examples of these in a moment. But the question is there any relationship between antibiotic exposure and subsequent disease risk? So I'm going to show you a study that I participated in with colleagues at the Mayo Clinic that was published about a little more than a year ago, looking at the association of infant antibiotic exposure with childhood health outcomes. Now, this was a study in Olmsted County, Minnesota, which has been the site of many studies over the years because it's a stable population. The people in Olmsted County are getting almost all of their health care from Mayo Clinic doctors. So they have very good records. So in this study, ultimately, we analyzed 14,000 children born in Olmsted County over about a 13-year period. We divided their records into a period of antibiotic exposure before they reached the age of 2, and a period where we then wanted to see what were their health outcomes going to be from age 2 up to age 13. In total of these 14,000 kids, about 10,000 kids had exposure to antibiotics in the first 2 years of life, about 4,000 didn't, and 3 classes of antibiotics accounted for almost all penicillins, cephalosporins, and macrolides. So here's the Kaplan-Meier plot of the time to developing asthma by sex and by antibiotic exposure. The x-axis is age. It begins at age 2. That's when our outcome period begins, and it goes up to age 14. And the y-axis is the probability of asthma. The red lines are girls, the blue lines are boys. So the solid red line are girls who did not have antibiotic exposure in the first 2 years of life. This is their rate of developing asthma. But if they had antibiotic exposure, this is their rate. It's about double. Boys, solid line here, dotted line with antibiotics here. Again, about double the rate. So I'm just using this as an example. In fact, we looked at 10 different common health conditions with childhood onset: asthma, allergic rhinitis, food allergy, atopic dermatitis, celiac disease, overweight, obesity, ADHD, autism, learning disability, inflammatory diseases, metabolic diseases, neurodevelopmental diseases. Here are the adjusted hazard ratios. A hazard ratio of 1 is neutral, no relationship between antibiotics and the later onset of these diseases. In fact, all 10 of these had hazard ratios greater than 1, and 8 of the 10 were statistically significant. And others, there have been many studies that have looked at single conditions. This is the first one that has looked across many conditions, and in fact has found very significant, consistent effects. We also found specific associations with the number of antibiotic courses, timing of exposure, and antibiotic class, and I'll show you that next. So here first, we're just looking, what I showed you was the composite of everyone, but if you separate it by girls and boys, in essence, we did 2 studies in parallel, and the results are quite similar. The odds ratios in girls and boys in general were quite similar to one another for a given condition. One exception is celiac disease, which had this very strong association with girls. But you can see in general the consistency. We're going to come back to autism in a moment. Here is the association between the antibiotic exposures and the cumulative probability of developing conditions. So the first panel are the kids who had no antibiotics, then 1 to 2, 3 to 4, and 5+. And you can see that the proportion of kids that has some condition is growing. It turns out that the number of antibiotics didn't affect whether they had one condition or not, but it did affect if they had 2 or 3 or more conditions. We can see this another way, that here's the amount of time it took to develop a single condition, and they developed their first condition by the age of 12, whereas these developed it by the age of 13. This is no antibiotics. But of course, the big expansion was with 2 conditions or 3 or more conditions. Here we're looking at the hazard ratio by timing of exposure. So for asthma, essentially all of the associations are significant, whether the timing is in the first 6 months of life, the second 6 months of life, or the second year of life. It didn't much matter. For allergic rhinitis, it's a little different between boys and girls. Very important in first 6 months of life, very important in the second year of life here. For food allergy, in both groups, antibiotic exposure in the first 6 months of life, that's where all the risk is. Atopic dermatitis, very similar to food allergy. I don't think anyone has seen this kind of data before. Celiac disease, no specific trends, but the numbers are very small for celiac. When we look at overweight, we see that the odds ratios are highest early exposure, and it goes down. And we see something similar in both males and females. This is very similar to what farmers have found. The earlier they give the antibiotics, the bigger the effect. We see something more or less parallel in obesity, but not completely. ADHD, no exact pattern. In these others, no particular pattern. Here we asked about the dose response. So here for asthma, we see really a clear dose response. The more courses of antibiotic, the higher the risk of asthma, going up to 3 in those who had 5 or more courses in females. Again, very similar dose response relationship in males. Allergic rhinitis, most of the risk is in people who have, in the children who had 5 or more courses. Atopic dermatitis is kind of in the middle. Celiac, hard to interpret. Overweight, again, there's a dose response. We see that. Obesity, there's a dose response as well. ADHD, there's a dose response as well. Now here we're looking at hazard ratio associated with exposure by antibiotic class. And this is some of the most interesting data. For asthma, it really didn't matter what antibiotic it was. Any exposure to antibiotic had a significant effect. Allergic rhinitis, the strongest effect was with the cephalosporins. Food allergy, cephalosporins. Atopic dermatitis, cephalosporins in the females. Celiac disease, it was the opposite. It was penicillin, had a very strong effect. Overweight, a little different in males and females, nothing very strong. Obesity, strongest effect, cephalosporins. ADHD, very strong effect of penicillin. Now, the most interesting result in the whole study was what we saw here for autism. And that was that cephalosporin in females, cephalosporins had a very strong relationship to the ultimate development of autism. And penicillin was inverse, as if it was protective. Maybe because if they were given penicillin, they weren't given a cephalosporin, or vice versa. And we see a similar type of trend. Again, cephalosporins were associated with the development of autism in both males and females. And again, these are essentially 2 separate studies. So this is a very interesting finding. And in fact, working with Manny DeSico-Bloom, we've now given cephalosporin to mice to see in an autism model to see if we can change the development of disease. So what about cephalosporins? Well, you know that there are 1st, 2nd, and 3rd generation cephalosporins. And over the last 20 years, what's come into the practice is there are these oral cephalosporins that can be given every 12 hours or every 24 hours. And for pediatricians, this has been a big advance so that they could only give antibiotics once a day or twice a day rather than every 8 hours or every 6 hours. So that's why cephalosporins have been growing in general in pediatric practice. But this study uncovered some new relationships. So up to this point, I've shown you evidence for an association of antibiotics and these diseases, but people take antibiotics because they're sick. So could there be a causal role for antibiotics? That's hard to study in humans, but we can study it in experimental animals. And I'm going to show some slides that I showed when I spoke to you last about 3 years ago, and then I'll show some new stuff. So the principle that antibiotics can change metabolism was established by farmers about 75 years ago. They found if they gave antibiotics to their farm animals, it promoted their growth. It works from chickens to cows, a wide swath of vertebrate evolution. It's true for essentially any antibacterial that's used, regardless of its chemical structure, class, target, or spectrum. Antivirals don't work. Antibacterials don't work. Importantly, the earlier they're started, the bigger the effect on growth rate, the bigger the effect on feed efficiency, conversion of food calories into body mass. That suggested to us a developmental phenomenon. So we began to study the phenomena in mice, in which we would give mice antibiotics or not, measure properties, analyze the microbiome, and look for a relationship. I'll show you a few of our many studies. Our first study was led by Il-Sung Cho when he was a postdoctoral fellow in the lab. Il-Sung gave 4 different antibiotic regimens at the midpoint of the FDA-approved rate for growth promotion on the farm, or no antibiotics, and measured the amount of body fat. And you can see that the mice that got antibiotics put on more fat. This was our first evidence that antibiotics might change metabolism. Il-Sung showed much more, but I want to go on to the next study which Lori Cox did when she was a graduate student in the lab. She asked, are the changes durable with only a limited antibiotic exposure? So in addition to mice that got no antibiotics or lifelong antibiotics, she had mice that got only 8 weeks of antibiotics or only 4 weeks. Here are the effects on phenotype, total mass, lean mass, fat mass. The black line is the control group. The mice that got the antibiotics, all the antibiotic groups had increased total, lean, and fat mass. So 4 weeks beginning early in life was sufficient for the full effect. So what was this doing to the microbiome? So here we're looking at fecal community structure in mice that are 3 weeks old. And at 3 weeks, there are only 2 groups of mice, control mice, no antibiotics. All 3 antibiotic groups are still receiving antibiotics at 3 weeks. In this principal components analysis, each circle represents 1 mouse. The black mice are the control. The orange mice are those receiving antibiotics. There's a lot of diversity in their microbiota in both groups. They overlap, but not completely. They're a little different. Not surprising. One group is getting antibiotics, the other is not. Now at 8 weeks, we have 3 groups of mice, no antibiotics, continued antibiotics, or 4 weeks of antibiotics and stop. So here's the controls in black. Here's the continued antibiotics in orange. They're more separate than before, not surprising. One group is continuing to get antibiotics, but the group that got antibiotics and stopped, their microbiota has normalized. So the effect of these antibiotics was transient, but the effect on the phenotype was permanent. And this was our first evidence that altering the microbiota during early life, during an early critical stage, could affect long-term outcomes. And in other studies, we have confirmed this hypothesis. So is it due to a side effect of the antibiotics, or does the antibiotic effect go through the microbiome? So to test that, we did a cecal transplant. Mice were given antibiotics or not. We sacrificed them. We harvested the microbiota from their cecal contents, and we gave them to germ-free mice. These mice never saw an antibiotics over 5 weeks. And now we're measuring what happened to their total lean and fat mass over these 5 weeks. What we found is that the mice that got the antibiotic perturbed microbiota had increased total mass, no change in lean mass, and increase in fat mass. So the metabolic signal is in the altered microbiota. We also were interested in immunological phenomena. So here we're looking at expression of genes involved in intestinal defenses in the ileum of donor and recipient mice. First, I'm going to show you the donor mice. We looked at Th17 genes, transcription factor, 2 cytokines, antimicrobial peptides related to Th17. In all of these, the mice that got the antibiotics, all these gene expressions were down. And that's what we expected from our prior work. So now we asked, what will happen in those recipient mice, the mice that received the antibiotic perturbed microbiota? And the results are quite parallel, where all these are down as well as the recipients. And this provides evidence that the immunological phenotype is also being transferred by the perturbed microbiota. Now that's one kind of model. The second model is work that Shusang Zhang has done in an animal model of type 1 diabetes in NOD mice that spontaneously developed type 1 diabetes. He gave these mice 3 courses of antibiotics at the equivalent dose that human children would get antibiotics, or no antibiotics. Or he gave a single course of antibiotics or no antibiotics, and then tracked whether they would develop diabetes or not. And here's a Kaplan-Meier analysis of type 1 incidence. The blue lines are the control mice. They didn't receive antibiotics, and they're developing type 1 diabetes as we expect in male NOD mice. But the mice that received antibiotics are getting diabetes earlier, and they're getting more of it. And even 1 course is sufficient for the full effect. We're going to come back to this model in a moment. And so this is further evidence of causal relationships between antibiotic exposures and disease phenotypes. Now, I want to take it 1 step further and tell you about studies in a model of IBD. The epidemiologic studies in Denmark show that the more courses of antibiotics that children get in the first year of life, the more likely they are to develop IBD, especially Crohn's disease. So we wanted to see, can an antibiotic altered microbiota affect IBD development? And to make it more interesting, we wanted to see what it affected in the next generation. So we studied wild type mice and IL-10 deficient mice. These are mice that spontaneously develop colitis. This work was led by Angel Schulfer when she was a graduate student in the lab. Angel gave either normal microbiota or antibiotic perturbed microbiota to germ-free mice that were pregnant. They were either wild type or IL-10 deficient. So she had 2 genotypes, she had 2 microbiota, she had 4 experimental groups. After conventionalizing these mice, the pregnant mice soon gave birth, and then we followed their pups into middle age. And we asked 2 questions. Does the antibiotic perturbed microbiota have an effect on the ecology of the gut? And the answer was yes. We showed it in many dimensions. You can see it in Angel's paper. And also, does it have an effect on the disease, on the IBD in that next generation? And I'd like to show you that result now. So here we're looking at colonic pathology in IL-10 knockout pups at week 21, according to the microbiota to which their mothers were exposed. Here's the colon of a pup whose mom got the control microbiota. This pup has colitis. That's what we expect in IL-10 knockout mice at age 21 weeks. But the mouse whose mom received the antibiotic perturbed microbiota, they have a huge amount of colitis. The differences are actually 30-fold. This is a representative difference. So there's much more disease in these. So let me summarize this experiment by reminding you that neither the pups nor their mothers received an antibiotic. They never saw an antibiotic. That means that the enhanced disease signal that we're seeing here is entirely microbial. That means that antibiotic effects can cross generations. And it also means that inheritance is not just based on host genes, but it's based on microbes and on their genes as well. So to summarize what I've said so far, antibiotics have long-term effects on host metabolism and immunity. The effects are due to perturbing the microbiome. Other effects of modern life also contribute, but I haven't discussed them. The effects may be transmitted to the next generation, and it's clear that we need to find and implement solutions. So where is the hope? Well, there are 2 kinds of solutions. The first is that we must steward antibiotics better, and this is a really important area that Keith is very involved in. We have to curtail unnecessary use. But even if we do that very successfully, we'll still be where we are. We actually have to ultimately restore microbiota. So Shusong Zhang continued his studies of type 1 diabetes. He had a control group of mice that were just NOD mice and didn't get any antibiotics. He had a group that got antibiotics, and then he got a group that got antibiotics and then were gavaged by cecal contents from moms. So we tried to restore here and see what would happen to the diabetes. So here's the Kaplan-Meier curve. The blue line are the control mice, no antibiotics. Red line are antibiotics, and the green line are the mice that received antibiotics but then got cecal contents back. And we brought them back almost to the baseline. So this provides evidence that we can restore even after the early life perturbation. Shusong showed many things in this study, which was recently published in Cell Hosted Microbe. Here, I'm just going to show you one example. We're looking at gene expression in the ileum of the mice in relation to the restoration. So this is a heat map based on RNA-seq data of the mouse ileum when they're 23 days old. This is unsupervised hierarchical clustering. Each column is one mouse. So what we found is that the mice that received the antibiotics cluster all by themselves. Here are the control mice, they're clustering by themselves, but they're also clustering with the mice that received antibiotics, but then got the cecal transfer. So their microbiome more resembles the controls than other mice that received antibiotics. And in particular, we can hone in on particular genes in which the pattern is clearly control in either direction, whereas there are intermediate groups as well. So we know what all these genes are. So this is a pathway for discovery of microbes, microbial genes, metabolites, and host genes that are perturbed by antibiotics that drive autoimmunity and that can be restored. And this is something that we're working quite accurately on. So let me just finish up by saying presented evidence that there's diversity loss in the microbiome. We can think of the world as having three model locales: a developed country like the United States, where we began to modernize very early and diversity has been declining; an intermediate country that began modernizing more recently, like India or China, but they're catching up in a hurry. And then this might be countries in Latin America or Asia or Africa, where it's just modernization has just begun. This was my view in 2016. And the question is, what's the future going to bring? Is our microbiome going to decline further in diversity? Is your generation going to arrest the decline? Or perhaps we're going to reverse this through restorative steps. And these restorative steps, part of it is restoration, but we may have to remove certain opportunistic pathogens as well. So there are many needs for research in this area. So I believe that the medicine of the future will include a new analysis of child health. The pediatricians of the future will examine babies and their diapers. They'll analyze the baby to see, does this baby have the global microbes that all babies need, or personal microbes that a baby of this genotype should have? And if they don't, they will reach into their armamentarium and administer those missing microbes, and ultimately study whether this has optimized their health, and if necessary, continue to reshape the early life microbiome. If microbes are disappearing, where will they get them from? Well, we believe that we will get them from the Microbiota Vault. This is a global nonprofit effort to store disappearing microbes before they disappear. It's being led by Maria Gloria Dominguez. Rutgers is part of this project. There's an international foundation. You can look at our website, www.microbiotavault.org. I also just want to go back and recognize the many people who did the work I've shown today. This involved people in the lab. I've tried to indicate them as they went along, but many different collaborators and funding over the years from foundations and from the US government. And with that, I will stop and see if anybody has any questions. And I'm going to stop sharing my screen.
Marty, that was wonderful.
Yeah, I had a question. Thanks for that amazing and absolutely frightening talk. I had a couple. So first, you mentioned, that was interesting, the data that you showed on that bioessays about the cultural drivers of antibiotic prescriptions, and talking about religiosity, masculinity, things like that. Has anybody applied that type of study to physicians themselves? I think about specialties that we deal with in the hospital that we struggle the most with antibiotic stewardship, and often have some of those same characteristics, you know, ICU surgical specialties that seem a little more black and white thinking, a little more masculine. So just that was one question. And then and I can wait on the second question.
Well, let me just say that I'm glad you found it frightening, because I do too. And that's why I'm speaking about this, because we could ignore this, but we're just digging a deep hole. And until we get on top of this, the hole is going to get deeper and deeper. That's why I call it climate change in miniature, because it's the same kind of phenomena, something very slowly advancing, where we're changing our ecology. Climate change, we're changing our macroecology. Here, we're changing our microecology. So, you know, I think that's a really important issue. And it is frightening to me as well. Second question is, has anyone applied this to medical practice? There's been some work in Europe, but I think that there's a great opportunity for scholarship there to try to link antibiotic utilization data with personality and psychological characteristics. The evidence, it seems to me that it's not about the year that people graduated from medical school. It's not necessarily whether they're male or female or many other factors. It seems to me that it has a lot to do with personality and personal psychology. And I think that's one of the keys. Patients find doctors who have the same kind of personality and psychology as they do. If they want antibiotics, they find a doctor who's going to give it to them. If they don't want antibiotics, they find someone who don't. This is at least as outpatients. In the hospital setting, we probably see something similar, but the dynamic isn't controlled as much by the patients. So I think this could be a really interesting area of scholarship. I agree with you.
Marty, this is Keith. Just to sort of follow up on John's question, are you collaborating with, or do you know of any sort of sociology people or folks who are digging deeper into this from, sort of a non-medical perspective, but more of a behavioral and sociologic perspective?
People are beginning to look at this in terms of antibiotic stewardship, as well they should. I'm not sure that anyone has approached it kind of as directly as our work implies. And that's one of the reasons we wrote that paper. That paper is me and 3 anthropologists, because they added a lot of value to that study. And I really recommend it. That paper's got like 250 references to papers that you've never seen before. So it's quite rich. In fact, I can send that paper to you, Keith, and you can distribute it to everybody if they like.
I would love that. Thanks so much.
I think it's a really important frontier about stewardship, not just in the hospital, but again, 80% of the antibiotics are used in the outpatient setting. That's where the big problem is. That's what's driving resistance. It's not, it's also being driven in the hospital, but the big reservoir is people in the outpatient world.
I think that outpatient world is, as hard a nut as the hospital is to crack. The community is like the ocean. It's almost overwhelming in terms of controlling a really stewarding antibiotic use. So I hear you.
Well, one thing to say is that, again, the community is heterogeneous. You saw these tremendous regional differences. There are also differences by types of prescribers. Per capita, nurse practitioners and PAs prescribe more than physicians. Doc in the boxes prescribe more than doctors in practice. In part, it's driven by the economics. It's driven by uncertainty avoidance. The doc in the box, they don't want any uncertainty, and they don't care about a problem 5 years down the road. They care about what's going to happen in the next 24 hours. So there, again, lots of opportunities to begin to look at this. There's a wonderful scholar at the University of Pennsylvania. I can't remember her name, but I will, and I'll send that to you, Keith, who's beginning to look at some of these issues.
Is that Julie? I think that's Julie Smythe.
That's right. That's why I can't remember her. That's why I can't remember her name.
It's an SMY and lots of consonants, but yeah, she does.
But her first name is Julie or Julia. She's doing really fantastic work.
Marty, what about the drug companies? They're obviously in this in a big way and don't like to hear things that you are talking about, since it will surely affect their bottom line.
Yeah. So our paper talks a lot about the economic influences on antibiotic prescribing, and there are a lot. Let me just point out that every time an antibiotic is taken, money changes hands. And one of the things I learned from our study, many of you have heard about in China about the barefoot doctors that was initiated about 50 years ago to bring kind of healthcare to rural areas, and has been considered to be an enormous success. What I learned more recently is that now that China is getting more capitalistic, the compensation for barefoot doctors, these health workers in the rural areas, is tied to every time they prescribe an antibiotic, they get a bonus. So they have a financial incentive to prescribe antibiotics. Also in China, hospitals, every time an antibiotic is given intravenously, they get a bonus as well. Again, it's based on the idea that antibiotics are wonderful. So that kind of makes sense, except when you look at all the effects. So the big drug companies, they're not, as you know, they're not that interested in antibiotics anymore because these are short term. They're more interested in lifestyle antibiotics, except for high ticket items like advanced spectrum cephalosporins and azithromycin, which is, I think, the most profitable antibiotic in the United States. When I went on some expeditions with my wife to South America, when we went to even the smallest villages, we would see little bodegas selling generic over-the-counter antibiotics. They're almost all manufactured in China. China is manufacturing about 250,000 tons of antibiotics a year. The United States is manufacturing about 18,000 tons, just to give you a sense of the scale. A lot of it is used domestically in people and on the farm, but a third of that, 80,000 tons of antibiotics, are being sold to other countries in the world, mostly for use on the farm, but plenty for use in people. Well, I'm happy to have stimulated some thought. I would like to send you that paper for you to kind of look in depth, really about the sociology of antibiotic using. Wherever you look, there's variation in antibiotic use, and that intrinsically tells us that there's something wrong.
Marty, thank you so much. I look forward to distributing those manuscripts. So thanks so much. It was a wonderful talk.
Thanks, everybody.
Great day. Bye-bye.
Bye-bye.